Hi, everyone.
Hi, Greg.
Andrew.
Thanks for giving the talk today.
My pleasure.
We'll just give it another couple of minutes while people join.
Hey, Greg.
Hey, Jim.
How's it going?
Good.
I heard there's a talk today, so that's good.
What's that?
I heard there's a talk today.
Yeah, me too. I just finished my slides.
Awesome. Andrew, did you want to do the introduction?
Sure.
Okay. I think we're at 1 o'clock and we got a pretty good attendance already. So why don't we get going?
Okay. Yeah. So thank you for joining the BBSR seminar for this month. Some quick housekeeping. This will be our last seminar until September. So we'll take July and August off. So the last one of this fiscal year will be Greg Way, who is a relatively new assistant professor. He's a part of the Center for Health Artificial Intelligence, or CHI. And today he'll be talking to us about learning biology from microscope images of cells. So Greg, I think we're all excited to hear your talk, and you can get started.
Sure. Can you see my screen? I feel like you can't.
No, we could before. Now I just see terminal Python.
That's the right one. Perfect.
Yeah.
Awesome. Okay. Well, thanks to Andrew and Jim for the invitation to speak at the seminar series. I'm looking forward to presenting the types of work that my lab does. So like Andrew mentioned, I'm a relatively new faculty member in the brand new Department of Biomedical Informatics. And the title of my talk, like Andrew mentioned, is Learning Biology from Microscopic Images of Cells. And so I'm going to split this talk up into two parts. It'll be about 50 minutes, hopefully. And I'll pause for questions after the first part, which is kind of a brief introduction to the data type. And then in the second half, I'll talk about two different applications. So if I have such a broad title, I think it's probably best for me to start from a long time ago. So you've probably all seen this quote by Isaac Newton. You see it every time you open up Google Scholar, some version of it. If I've seen further, it is by standing on the shoulder of giants. It's a pretty popular quote, and I'm sure you've seen it before, but I think a little known fact is that the origin of this quote was in a letter that Isaac Newton had written to Robert Hooke, who was the father of modern cell biology. He was the first person to focus a microscope into living creatures, and he saw these things that he thought looked like cells that monks would live in a monastery, and that's where cells come from. So we've been learning biology from microscopy images since Robert Hooke in 1665, so over 350 years ago. But now is quite a lot different than the mid-1600s. I think some pretty important pieces about how now is different. We have computers. We have the ability to store data, and we can analyze data really fast using advanced algorithms. And so obviously these images here aren't reflective of exactly the advances that we've had in modern times, but there's been much development in this space, and the pace at which we're making these developments is pretty incredible. But I think one key difference between now and then is that we have a much better understanding of biological complexity. In Robert Hooke's time in the mid-1600s, he wasn't at all, he had no clue what he was looking at. He didn't understand that there was so much embedded in the images that he was looking at. And one of the major reasons why we have this biological complexity is, in large part, a culmination of many, many years of research that could be in some ways summarized with the completion of the Human Genome Project, which completed in the early 2000s. So this project really demonstrated the team science that's required for such a complex endeavor that faced loads of technical challenges and continues to have immense impact. More recently, there's been the development of the Human Cell Atlas, which is looking at cells and different cell types, trying to catalog different cell types and cell states in a variety of different contexts. And from these efforts and many, many years of biological understanding, we have come to the central dogma of biology, where you go from DNA to RNA to protein, and all of that function exists within a cell. But what's key about all of these things is that if you measure one of those aspects of a cell in one specific way, whether you measure the DNA or you measure the RNA, you might not find that signal in another data type. So this is a well-known characteristic of the central dogma. If you measure mRNA abundance, it's not going to strongly correlate with protein abundance. So if you measure a gene expression, it's not necessarily going to coincide with the amount of protein that's present. So why then study biology from microscopy images of cells? Structure equals function in biology, and with images of cells, that's really close to structure, and therefore is going to inform a lot about function. Images are also really cheap and fast to collect. You can snap a photo with a microscope relatively easily. The data are typically pretty large to store, but it's really fast, and you can collect a ton of samples. There's also no need to destroy the sample like in molecular readouts. You can collect live cell imaging rather easily. And what we're finding is that, like RNA and protein, the readouts that you're deriving from microscopy images are complementary to other approaches. And so there's two broad categories of how people typically think about learning biology from microscopy images. These are targeted and unbiased. And so just as a quick example, both of these categories are often combined in the same experiment. So you could have a targeted image assay where you have some molecule that's tagged with GFP, and you can visualize the localization of that particular molecule using this GFP fluorescence reporter. There's also unbiased categories where, for instance, in the same experiment, the authors have also tracked MitoTracker. So this is a generic stain that's going to report on the localization of mitochondria in general and the distribution of an organelle that's really important in a lot of biological function. And so I'm going to focus on the unbiased approaches to learning biology from microscopy images. I think that this is really where we're going to learn a ton of information about disease and drugs. So this is kind of just an example of the types of images that my lab studies. This is a cell painting image of various dyes that are targeting unbiased organelles within a cell. So I'll talk in much more detail about this assay, but just to get a sense of the kind of data that we work with. And so cell painting is one example assay that we analyze. And you can think of cell painting kind of like a color by number, where you select different organelles to color with different reagents that are going to be along different fluorescent spectra and are going to emit at different wavelengths so that you can really easily capture it in a multiplexed experiment. So the traditional cell painting assay is going to stain for these specific cell compartments. So we have a nucleus channel. We have a channel that targets endoplasmic reticulum, nucleoli and cytoplasmic RNA, actin, Golgi, plasma membrane, or AGP, as well as mitochondria. And so typically when you see the cell painting assay, it'll be merged into a single representation across the different channels. But know that these are individual reagents that we're using to stain independent cell painting compartments. The cell painting assay is also really customizable and not really all that much optimized. So there's lots of room to improve the assay and lots of flexibility to be creative. We also study phase contrast imaging. And so this is a dynamic data type that exists in time, as well as XY dimensions. So you can imagine studying the process of mitosis and how cell morphology changes across the different cell cycle stages. And you can overlay perturbations with this, knock down genes to see the effect of how the gene function is impacting different stages in mitochondria. So now I'll get into a little bit more of the bioinformatics side of the data that we work with. So there is kind of a standard experimental workflow that we typically think about. And all of these steps are very modifiable. And it's very flexible to study a whole bunch of different diseases or perturbations. So I'll just walk through these next couple of slides and I'll kind of narrow in on the kind of where my lab sits in terms of the methods that we're going to develop, but also just like a standard workflow to see if anyone in the audience might be interested in applying these sorts of workflows to their own particular problem. So we start with cells. These could be healthy cells, diseased cells, cell lines, or patient-derived lines. Optionally, we perturb them. We do this pretty often with drug panels for phenotypic drug screening or genetic perturbations. So this could be like CRISPR or RNA interference. We plate these cells on 384 well plates or 96 well plates, and we apply high throughput staining. So we typically the cell painting assay, but like I said, that's modifiable. And so from this procedure, now, if it's a multiplex imaging assay, we have, we receive images, we apply a microscopy, confocal microscopy to extract images of these cells. And we perform an image analysis and feature extraction pipeline to convert to essentially segment the cells, extract features from these cells, and convert the images of the single cells to a feature matrix, and then perform downstream discovery. So this feature matrix, I feel like probably most folks in this audience would be familiar with this kind of data as they study a whole bunch of different disease contexts in biomedical contexts. Like you could use this matrix for like you would a gene expression matrix or a matrix of protein abundance. And so the bioinformatics really sits in this space here of actually performing the image analysis and performing the feature extraction, as well as processing this downstream matrix. And so now to go a little bit deeper into the bioinformatics, this is another view of that's focused on the data. So from a data-centric perspective, we start with the rawest form, which are the images that come off of the microscope. Each of these arrows represent various more or less standard bioinformatics pipelines that are used to process one data type to convert it into another. So for instance, this QC pipeline, this typically involves removing debris or removing out of focus images or trying to adjust for them, and correcting for background intensity or illumination correction. So we take these microscopy images, we perform a QC pipeline to adjust them and make sure that we're analyzing the cleanest possible data and removing all known artifacts. And from there, we then can apply, we have a couple of decisions to make. We can apply representation learning pipelines, which I'll talk about in the next couple of slides. And then we also most commonly perform a segmentation pipeline to identify the boundaries of the cells. And from there, we get these masks that are output, which are going to distinguish background from foreground. And then there's an optional step here, which I won't discuss. But essentially, this process of representation learning extracts these morphology features from the images. And we get, essentially, that's the conversion to this matrix, where we arrive at single cell morphology embeddings that we then apply an additional bioinformatics pipeline to further refine the signal that we're receiving from these data. And this is a rather new approach. So within the past couple of years or so, and there haven't really been a lot of really great solutions for data storage. The data are typically large, anywhere between 500 gigs to 40 terabytes and higher. So there's quite a lot of considerations that you need to make when approaching and beginning an experiment to make sure that you have proper data storage policies in place. So I'm going to talk more about this representation learning pipeline. I'm also going to talk more about this bulk pipeline. These two, I feel, are probably the most ripe for innovation. All of these others are more or less fixed in place, and there's still a lot of development that's going on. But my lab is most excited about these two bioinformatics pipelines. So just to discuss more about representation learning, one of the challenges in analyzing microscopy images of cells is there isn't this standard dictionary of features that you can extract from cells, like there is maybe for molecular readouts. So the Human Genome Project produced the sequences of all the genes in the genome. And so now we have those genes that we can hang knowledge on. We don't necessarily have that for cell images. We have things that are kind of close to it, but there's still lots and lots of challenges that is too much to discuss in this one slide. But one of the categories of representation learning that we use often is based on Cell Profiler. And so Cell Profiler is a tool that extracts these so-called hand-engineered features from images. So these are going to be things like geometric measurements. It's going to describe perimeter, area, shape-based features, texture features, granularity features, so how speckly are cells, the various intensity measurements, if you're looking at fluorescence microscopy, not only kind of the intensity of the stain, but also the distribution of the stain. And then also these other radial distribution-based measurements, which are kind of where the staining patterns are aggregated within a single cell. A more emerging data type is based on deep learning. And so this is Deep Profiler. It's kind of the spiritual successor to Cell Profiler. That is essentially the same paradigm. It's a drop and replace. But instead of using these hand-engineered features, it's going to use deep learning and trained models from TensorFlow. And this is where you can get really creative because you can train models using different data sets, and you can have different approaches and different architectures. But most often, we use a pre-trained ImageNet model in the context of Deep Profiler to derive representations. And so ImageNet is a...
Sorry, do you mind if I ask a question?
Please, go ahead.
So you might be talking about this in a little bit, but I'm curious on how good the prediction is across different cell lines, in particular ones with pretty significant morphological differences.
What do you mean predictions?
Well, identification of features, right? So how does the quantification look across, more mesenchymal, like spindly versus more cuboidal? How did those, I'm assuming those were in the training model. I'm just kind of curious on how the actual biology of the cell affects that.
Yeah, so that's a great point. I mean, you can look at a single gene and that gene's expression is going to have relatively similar function across different cell types. But if you look at the cell morphology, it's going to be likely pretty different if you're looking at very different cell types. These features are pretty consistent. So like perimeter and area, those are obviously going to be different because they're like more traditional morphology features that are going to directly report on the differences. But these intensity-based metrics and these radial distribution metrics, a lot of them, what they do in this case, there's a transformation step that kind of takes the different shapes and morphs it into a common shape and then makes the calculations from the common shape. We do have, I will talk about one example later where we have multiple cell types and machine learning models that are trained to predict different higher order phenotypes like DNA damage. If you apply those models to different cell types, in a lot of cases, they translate. So I think that's evidence that the features are more or less consistent, at least some of them. like the areas and stuff, those are going to be definitely different, but at least for cell profiler. Now, deep profiler, we don't have as much evidence to suggest that different cell types are going to have similar features for the same deep learning model. I suspect that they will for many of the case. Like, I think it's going to be pretty similar to cell profiler.
Cool. And then do cell actual, that might not be a huge factor because most cells are pretty similar in size, but there are cells that are much smaller and there's cells that are much bigger. And then does the resolution have an impact on all of these models? I would assume it would, but.
Yeah. So I think that's a research question. The field is so new that we haven't answered that, or at least I haven't seen it answered in any way, really.
Okay.
Yeah, I think it's really interesting. Typically what we do in deep profiler, or at least what I've seen done in deep profiler, is that there is often like a resolution, like there's a resolution change that if you're analyzing two different data sets and one has a higher magnification than the other, I have seen a downsampling to make sure that they're on the same resolution. So I suspect that the features that you identify are going to be different depending on the resolution, and you probably want to keep it consistent. But to the extent that they're transferable, I think it's still unknown.
Cool. Thank you.
So after the representation learning step here, we derive these single cell morphology embeddings. And then there's a whole host of other processing steps that is typically performed to go from single cells to bulk. And so this is a tool that the lab is developing right now called PyCytoMiner, which is containing all of the functionality to go from these single cell representations all the way through the typical image-based profiling pipeline, which is the process in which you transfer single cell data into bulk profiles. And so this is if you're studying in like a 3 to 6 well plate, a 3 to 4 well plate, or a 96 well plate perturbations, and you can get signatures for each of the individual perturbations. That's typically this paradigm. And so I'm not going to go through all of the tools. We're actively developing this. It's essentially it's a bioinformatics tool that has a series of different functionalities that are common in the field. And we hope to improve all of these individual steps as we go through the different data processing steps. So things like normalize, historically, these haven't really been all that explored. So we're hoping to develop different normalization procedures, different feature selection procedures, different batch effect correction methods, and so on. And so just to reiterate why one might want to do this is that this process is able to extract a systems biology readout of cell state. And so kind of just this one slide example of what we can do downstream. So this is work led by Ted Natoli and Adeni Adeboye, in which we profiled over 100 million single cells from A549, which is a lung cancer line, only one cell type. We applied 1,327 different drugs across 6 doses, and we measured them in about 253 to 4 well plates. And we used this exact same plate map layout to collect both gene expression and cell morphology data via the L1000 cell painting assays, respectively. We performed the traditional processing pipelines and got matched L1000 and cell painting data for all of these perturbations in A549. And so this is a UMAP representation of the cell morphology data, in which every single data point represents an individual perturbation and replicates of that perturbation. And you can see that many of the drugs are moving away from this blob of DMSO controls, which are the plus signs. There's not a ton of diversity here. So it's not like what you see typically in like a single cell RNA-seq UMAP, where you have a bunch of islands that represent the different cell types. But remember, this is a single cell line, A549, perturbed with a whole bunch of different categories of drugs. So these are in green, these are proteasome inhibitors. at fairly high doses. These tend to be very disruptive in cells and therefore cause very distinct cell morphologies, and a whole bunch of others that we chose to highlight, which ended up having fairly high signal in our data. And because we had matched L1000 data with the cell painting readouts, we can train machine learning models to predict the compound mechanisms. So I should note that we didn't just randomly choose the drugs. These were all drugs that were annotated fairly robustly through the drug repurposing hub effort. And the drug repurposing hub is a collection of clinically relevant chemical compounds, which are either FDA approved already or in some level of the clinical pipeline. And all of these have, not all of them, but many of them have pretty reliable mechanisms of action annotations. And so what we did was we trained the same machine learning models, the same architectures on using the L1000 data and the cell painting data. And this, each of these axes represent how well our models were able to predict each of these mechanisms of action. So you can see most of the mechanisms of action we weren't able to predict at all. So either they had too few examples in the training set, or they just didn't have consistent enough signal in the data that we collected. But anything below this dotted blue line, we were able to predict better using cell painting readouts than L1000 readouts. So things like PLK inhibitors, P38 MAP kinase inhibitors, dehydrogenase inhibitors were predicted much more robustly using cell painting. But then there were a handful of MOAs that were predicted rather well no matter the assay. So this is the conclusion of part one, just a summary of the introduction. So scientists, we've been studying microscopy for a really long time, since the days of Robert Hooke, but there's been a lot of progress that has happened over the course of these 350 or so years. We've built software and tools to analyze the data, and we have an immense amount of compute that's ready and able to really start churning on these data sets. Fluorescence microscopy and also microscopy in general provides advantages to molecular readouts. I might argue that it's a readout that could be a little bit closer to function. There's no need to kill cells for the live cell imaging cases, but there's also definitely some disadvantages. For one, cell morphology features are way less well-defined than molecular features. But nevertheless, they provide a systems biology perspective. So I'll pause there and see if there's any questions.
I have a question.
Great.
I'm curious if you know about the merging of the individual fluorescence channels into a single color image. You showed 5, if I remember right. And to my naive understanding, it would seem like you're losing information going from 5 individual channels to 1 color channel, which has just 3 channels in it.
Yes. So we don't analyze this merge representation. We analyze these 5 separately.
And then the
this
merge
is just
for
it's just for like writing papers and putting nice figures in grants and stuff. Yeah.
Thank you.
Thanks for clarifying. Maybe there's a question in the chat. Oh. So from Janani, regarding part 1, comparing drug families, any patterns with respect to what kinds of drugs are predicted better with cell painting and why? Yeah, so that's a good question. We didn't really dive, I don't think I have like a great answer actually. We didn't dive too deeply into the categories of compounds. We did some like very preliminary surface level, like seeing if antagonists were predicted better than agonists or inhibitors versus activators, and we didn't see any difference. So yeah, it's hard to say at the moment, but all this, I should say all this data are publicly available, all the code, all the labels. So we hope that people take this and do these kinds of, and answer these kinds of questions. Okay, cool. Okay, so I think I'm right on time. I'm going to talk through 2 separate applications of how that I think demonstrate how we're thinking about how this data could be used. The first one I'll go through is an example of predicting cell health. So as a motivation, it's easy to think about, looking at a cell and wondering what's going on. There's tons of things. If you ever look at like these electron microscopy images, they're so densely packed with stuff that's happening. It's like crazy to think about what we would need to do to figure out what's going on inside of them. They're much more complicated than these plush toys. And this is another representation that might be closer to truth, where this cell is kind of nasty looking. It's sticking all over the place. It has these projections. Maybe it just divided. It looks like an angry looking cell. What would be great is if we could use computer vision to isolate the cell and then use its features to determine what's going on under the hood. Like this cell kind of has a pretty high probability that it just divided. It might look like it's actively proliferating and trying to spread out. It's probably not in G1 or G2, and it might have a certain amount of reactive oxygen species that's going on. And it also kind of looks like it has high DNA damage. This is obviously an example, but it would be really cool if we could go from this kind of resolution data, which is much cheaper to collect and might not provide as much information, or maybe work on our microscopes to be able to collect more higher resolution images, but to be able to perform this type of detection to determine what's going on under the hood. So to get into answering this question, we actually had to design a separate assay, and I'll walk through kind of the experimental approach in a little bit, but I'll talk through, and this is going to also demonstrate the power of microscopy, because we can design these other orthogonal assays. This was in collaboration with Maria Costa-Alamova. She was my co-first author on this paper, and she designed this assay, which we're calling the cell health assays. So these cell health assays are collected in two separate panels. The first panel, we collect EDU, which is going to mark S phase cells, PH3, which is going to mark mitotic cells, gamma H2AX, which is going to report on DNA damage, and Hookst, which is going to denote cell boundaries. And so Masha is performing an image analysis pipeline on these data to essentially a series of semi-automatic gating strategies to separate non-round large cells or regular cells, round large cells. She's going to isolate cells that are in G1, G2, or S based on a combination of EDU and PH3. And she's also going to use image analysis tools to count the amount of DNA damage and identify mitotic cells. And side by side to the cell cycle panel, we also developed a viability panel, and both of them together represent the cell health assays. And so inside the viability panel, we have 3 separate reagents. Caspase 3,7 dye, which is going to mark apoptotic cells, Drac7, which is going to mark dead cells, cell rocks to measure reactive oxygen species, and we use bright field for segmentation. And so this is an example of the complexity of the image, even in a single cell line, with, one might think is rather homogeneous. There's really quite a lot going on. You have these exploding cells with very high DNA damage. You have cells throughout the different stages of the cell cycle. There's quite a lot going on. And so I already described the cell painting assay, but just to note that we collected the cell health assay and the cell painting assay side by side, much like we did with the L1000 and the cell painting data side by side across the same perturbations. In this case, they were CRISPR perturbations, but that's not really important for our application. Just to note that the goal is to use the cell painting data as essentially an unbiased readout of cell state to predict the specific targeted reagents of the cell health assay. Also to note, we used cell profiler data in this particular example. So just to make what I said more concrete, we use the cell painting data as our X matrix. We use our cell health readouts, which are more targeted readouts, plus Masha's semi-automatic gating strategies as our Y matrix. We fit regression models. So these are logistic regression models. And essentially the goal is to use this X matrix to predict the cell health phenotypes and by fitting regression models. There are 70 different cell health phenotypes, each reporting on different measures of cell cycle or DNA damage, reactive oxygen species. And so our hypothesis here is that we can use these unbiased reagents, that they capture enough signal to be able to predict the very specific cell health reagent readouts. And so I'll talk through two different aspects of the results. First, just going over model performance. In this data set, we collected 119 different CRISPR perturbations targeting 59 different genes across 3 different cell lines, A549, ES2, and HCC44. And so these are the results. This is in the test set, so data not used during training. The longer the bar, so the further the bar is on the x-axis, the better the performance. And these are each of the 70 different cell health reagents that we fit individual logistic regression models to predict. And so you can see the very first highest performing model, because we measured the actual shape of the cells, it turns out that we can predict it pretty well using cell painting data, which is not surprising, kind of like a positive control. But you do see that there are some differences across the different cell lines. We can also predict the number of live cells really well, the percentage of dead-only cells. Another measure of roundness, the number of cells that are in S phase, that are cycling in general, the percentage that are alive, the percentage that are dead, the intensity of the EDU stain in the nuclei, and so on. But you can also see once we start getting from the top handful, the lower performing models get really bad and essentially have no signal. And as you continue to move down, there's a lot of variability that it's induced in the different cell lines. One limitation in this study was that we had only 119 guides by 3 cell lines. That's 357 different perturbations that we were training our models on. That's a really small data set to try to fit a machine learning model. In the paper, we have a figure where we slowly remove samples and show that by no means have we plateaued in our performance, which indicate that if we increase the amount of data, we're likely to also see an increase in model performance. This is a figure that's summarizing the previous figure, which is categorizing the different cell health phenotypes on the bottom here. So we can predict cell viability based phenotypes really well. We can predict death phenotypes also equally well. These can kind of be categorized in the same group. We can predict S phase and G1 and G2 phase also fairly well. Mitosis is a little bit worse. Shape is kind of has high variability, but you can see that there's quite a decent performance for many of the cell health models. Others are pretty unreliable. These are just a couple examples of like the actual scatter plots of the predictions that really demonstrate like the lack of sample size that we have. So for S phase cells, we in the training set we can predict it pretty well, which translates also well to the test set. We also have this compared to permuted data, in which we randomly shuffle the feature matrix and then retrain, and we can show that we lose predictive signal in the permuted case. What was really exciting to us about this type of approach is that now that we have these models, we can use transfer learning to apply these models to different legacy cell painting data sets. So we actually applied it to the same data set that I talked about in the previous section, the perturbations from the drug repurposing hub. And so this is just 2 examples of 2 moderately performing models, the predicted number of reactive oxygen species and the predicted G1 cell count. And if you apply these models that we learned using the CRISPR data set to the drug data set, we can see that it validates for these 2 cases, at least in for a handful of compounds. So all of these red points here in diamonds and triangles represent the proteasome inhibitors, which we know induce high amounts of reactive oxygen species in A549. And these larger triangles here in blue represent PLK inhibitors, which we know their mechanism is to restrict exit from the cell cycle, indicating that they would have a lower number of cells that are in G1 that have just exited mitosis. All of these circles here are the DMSO controls. Furthermore, if you look at one specific PLK inhibitor, and I think others as well, but I just showed one example here, we see a pretty strong dose response as well. So as you increase the amount of this compound that's present in this set, you get kind of an expected dose response curve. As you increase the dose, you get fewer cells that are in G1. This is just a note to show that all of this, all these analysis are fully reproducible on GitHub. All of the data are publicly available, versioned and cited. And we also have an exploratory web app that you can go to explore all of the drugs that are in that are profiled by Cell Painting in the Drug Repurposing Hub across all 70 of the different cell health models. This is a work that was in collaboration with Shantanu Singh, Paquita Vasquez, and Ann Carpenter at the Broad Institute, along with co-first author Maria. Okay, so I'm going to jump into a second application now, but maybe it's also worth pausing to see if there's any questions on the first application.
Yeah, Greg, what's the landscape of people generating this type of data or the ability to generate this type of data on campus?
I haven't figured that out yet. I'm starting to. I know we have a microscopy core, and I've talked to the folks there a couple times. To be honest, it's been kind of tough to acquire collaborators and generate excitement about this data. And it's possible I haven't reached out to the right people. But yeah.
Are there specific types of questions, or, I mean, the drug repurposing stuff that you mentioned, but other types of questions that you're particularly interested in? I'm just giving you an opportunity to.
Yeah, definitely. So I think my second application is going to be more about like my current interests. And so I'll definitely talk through that. But yeah, thanks for the opportunity to talk about that. We applied for the AB Nexus, Sabrina Spencer and I. That was an extension of the second application that we're going to talk about. We're still very much interested in pursuing that work. It wasn't funded. There's also several other really interesting applications that we can maybe connect about later on, and maybe you can point me to the right folks.
Sure, absolutely. Okay.
Okay, so the second application is one in which we can use these kinds of data to predict drug resistance. And so just some motivating text. 25% of cancer patients benefit from precision medicine. You know, this is a really high number from a couple of years ago, but still relatively low. We want more people to benefit from precision medicine. We've made tremendous amounts of strides in cancer survival rates over the past 5 years, where these pink dots represent the 5-year survival from 1970 to 1977, and the blue dots represent about 40 years later. And so in almost all cases, with the exception of uterine cancer, we've dramatically improved 5-year survival. But what do we need to do to bump that number even more? And I'm sure that we're doing it actively right now on campus and elsewhere. But one really important piece of this whole puzzle to increase 5-year survival and also survival beyond that is this challenge of that tumors eventually develop resistance to therapy. And so a question is, if we can identify resistant cells before treatment, perhaps we'll have some understanding of treatment success, or even try to design combination therapies to better personalize a therapy for an individual patient. And so the question that we asked was, if we used this model system to see if this was even a feasible approach. Can we distinguish cultured cell lines that are sensitive or resistant to bortezomib? So we used colon cancer cell line HCT116. In this wild type parental strain, this is a known microsatellite and stable cell line that eventually generates many clones that are resistant to bortezomib. So if we treat it, we can identify these colonies that are resistant to bortezomib, and we can proceed with resistant colonies and non-resistant colonies through single cell cloning to generate a bunch of different replicates, biological replicates of the parental line. And so just as a test to make sure that our procedure is working, we generated 5, at least in this figure, we have since generated many more, but 5 resistant lines, plus these 2 historical lines, which we know have the specific mechanism is through a PSMV5 mutation in the proteasome subunit. But what this curve is showing is that these clones that we were able to isolate from the wild type parental line do exhibit the characteristic growth curve in the presence of bortezomib, indicating that they have resistance to the drug. In all of those clones, we applied the cell painting assay, and this is the 5-channel merge representation of the individual cells. And typically, if I'm giving this talk live, I would ask the audience if they could tell me which one of these images is resistant and which one isn't. So I don't know if anyone wants to jump out with a guess. It's kind of harder to do over Zoom. Yeah, so these 2 are the resistant ones. These are sensitive. So if you treat these cells with bortezomib, they will die. If you treat these with bortezomib, a low enough dose of bortezomib, they will live. It's impossible to tell by eye, but we wondered if we could train a computer to tell the difference between the 2. And so we use cell profiler features, and this is our approach. For each individual cell profiler feature or cell painting feature, we fit a linear model that is designed to adjust for different covariates: batch, treatment time, cell count, clone ID, and resistance status. So we really care about this particular covariate, indicator. And what we want to do is, essentially our approach was to select only the cell painting features that contributed to this covariate, to this status, rather than the other technical covariates. And so we did exactly that, and we identified a whole bunch of different cell painting features that were distinguishing wild type from resistant cell lines. More specifically, after we removed the features that also displayed signal with technical batch effects, we identified 45 individual features that were associated with resistance, and we applied these features in a machine learning framework to predict held out samples in a validation set, a test set, and a holdout set, which included clones that we didn't even include in the original model building exercise. And we found that we were able to rather robustly predict the resistance status, even in cell lines that the model wasn't trained on. So these are the features of the signature. This is like the typical cell painting, cell profiler based features. And you can see immediately that these are really hard to understand exactly what's going on biologically. Even people that have been looking at these features for over 10 years, they have a hard time explaining these. So that's kind of a challenge with interpreting these data. And I think that's essentially what I have for the applications. Yeah, so we can perform these predictions. It's difficult to interpret them, so we don't really know what they mean. Kind of where we're leaving off in this project. So as a summary, cell painting is unbiased and can capture important indicators of cell health without targeting any specific biology. We can use transfer learning across different cell painting data sets. We might have to consider the cell type, and it might not work in every cell types, but in general, we can use transfer learning to apply these models to predict phenotypes in different data sets. Also in the last example, we demonstrated that microscopy imaging detects patterns that are invisible to the naked eye. And perhaps in the future, we might be able to apply these types of signatures that are going to report on different resistance mechanisms to patient-derived lines to perhaps better predict and align therapy strategies. So in future directions, you can email me if this was interesting to you and you might have the capabilities of generating this data, or you want to work with me on a particular problem and I can find other people to generate this data. There's also loads of publicly available data. Our mission is to take, this image on the left, this is a microarray, and there are standard bioinformatics practices. No one ever has to look at these data ever, because there's so many common tools that are well established that can analyze this data and convert these images to gene expression readouts. We want to develop the tools that can enable just as easily people to analyze these data, because I think it's going to give you a lot of information that you're not going to get from this, and it's also amenable to other experimental designs that are going to be unique to the data type. And so with that, I'd like to thank everyone that was involved in various aspects of this project, particularly the folks that are highlighted here, my collaborators that I've been working with for a couple of years now. And thanks again. I guess I have plenty of time for questions.
I have a question.
Great.
You were using if you go back a couple of slides, the features that you were using in your analysis just then, these are from what software?
Cell Profiler.
Cell Profiler. And were those features not selected by hand?
What do you mean selected by hand?
How were those features? How does Cell Profiler work is what I'm asking. How does it get featured? What were those features and how were they created?
Yeah, I have a slide earlier here that describes this in better detail. Yeah, so Cell Profiler will look at a segmentation mask of a single image, a single cell image. It segments, well, you have to tell it to do it, but it segments the nuclei and the cytoplasm. And it's going to measure a whole bunch of these so-called hand-engineered features. So it's going to measure perimeter, area, these texture-based features like granularity. It's going to measure intensity across the different individual channels in the specified compartment. So if we go back to this slide here, so like cells, radial distribution, radial CV, ER204, that means that it was using the cells, like it's not just the nuclei or the cytoplasm, it's the whole cell. It's looking at this particular hand-engineered category of feature called radial distribution, more specifically this radial CV parameter in the endoplasmic reticulum stain. And this is another parameter as well.
So I don't understand how a feature can be hand engineered, but not understood. Those seem contradictory to me.
Yeah, so you can understand them, like you can understand what it's doing mathematically.
Okay.
I should have been more specific that like you can't understand what that mathematic descriptor is telling us biologically.
I see. Okay. Okay. Thank you.
But it's more, I guess, to kind of extend that thought, it's more about the combination of these features and trying to interpret what that means. Is that what you mean, Greg?
It could be, yeah. So, I mean, that could be part of the solution when trying to interpret these features. So it might not be worthwhile to look at one of these features independently. I mean, we know that in every data set that we analyze, like these features are very strongly correlated with one another. So they're likely reporting on similar aspects of biology. And there's likely some nuance as you go data set to data set. People try to analyze one feature at a time. And for some of these cases, like if you're a cell biologist and you've been looking at these features for a long time, a lot of them do tend to make sense, but also a lot of them don't. And it's still an open question.
Have you, I don't think I missed this, but have you tried to link, or have people linked genetic alterations or perturbations to some of these kind of feature sets?
Yeah, people have tried to do that. performed experiments in which because we have this links data set with matched cell painting in L1000, people have tried to predict one readout from the other using that same data set. It works to some extent, and many of these individual cell painting features are correlated to some genes, but it's not like perfect performance. It's not really like, there's some level of loss, of signal loss, when you try to go from one to the other, which is expected. But there hasn't really been too much in-depth effort on that.
Gotcha. Are there certain features that are just, every time you throw a drug on these cells, they're gonna get smaller or get more spindly or something along those lines? I assume there's some common response sets.
Yeah, I think so. I don't think people have looked at that. At least I haven't. I'm not aware of any. I mean, that's a good point. Like, there are some common responses that no matter what the drug is, at a certain high enough concentration, the cells are going to shrink, or even cells are going to lose adhesion to the plate.
Yep.
And so, like, that could be a pretty interesting project in which you try to identify a signature of that, and then anytime those combinations of features light up, that's telling you that this common response is occurring. So that would be cool.
Yep.
I have a question about, well, two questions. So first of all, I assume you're doing like 2D projections of these images. You're not doing like 3D analysis in Cell Profiler. And then the second question is, can these be modified to analyze like tissue as opposed to just individual cells?
The answer to the first question is most of the time we do a 2D projection from like some small range of Z-stack, but it's possible to do Z. It gets more complicated, and this like morphology analysis is much easier in 2D, so we tend to stick to 2D. You can definitely use Cell Profiler to answer your second question. You can definitely use these approaches in tissue. Segmentation becomes more challenging, and cell type annotation also becomes challenging. You might not need to do it in every case. The data are typically larger, and I found to be, I haven't really had too much experience analyzing tissue data, but I'm sure some of the challenges that we've overcome in 2D cell culture might translate to the tissue space. But I imagine there's also going to be a whole set of unique challenges.
Right. Okay. Thank you.
And just a clarification on some of the. So I think from everything that you showed, these are just general organ stains using the cell painter kind of kit. Is that correct?
For the two examples that I showed, yes, but we're definitely not exclusive to cell painting. There's other kinds of assays. We're particularly interested in live cell imaging, and there's live cell compatible dyes. We can just use generic bright field imaging, and I imagine depending on the tissue condition, you could apply these types of approaches also to tissue. But like I said, there's probably going to be a whole host of other challenges.
Yeah, I was thinking like the cyclic IF, or the Vectra, or the MIBI, or those types of assays. In addition, so with these, I'm assuming these approaches can be directly applicable to those data as well.
Yeah, I haven't played with it. I haven't played with that data, so I don't know exactly how applicable, but yeah, I know that the campus has a particular strength in generating those data, so it is probably worthwhile to explore.
Great. Anybody else have a question? We are at the top of the hour, so nobody jumps up in the next 3 seconds. And I think we're good. All right. Thanks, Greg. We appreciate your time, and it was very nice.
Thanks, everyone.
Thanks, Greg.
Great talk. Thanks.